for the talk. Let me hand over the mic and the stage to Professor Anand now. Thank you so much.
Yeah, thank you, Dr. Shukla, for that very kind introduction. So I'm going to go ahead and start sharing my presentation now. Can you confirm that you can see my screen?
Yes, absolutely.
Yeah, it's visible. It's fine.
Great. Yeah, so thank you again for that very kind introduction. It's a real pleasure to be speaking to you from Cleveland, Ohio. And I want to talk about the work that is going on in our group. I should also mention that not only am I a professor at Case Western Reserve University, I also have an appointment at the Louis Stokes Cleveland Veterans Administration Medical Center, where I'm a research health scientist. And so the work in my group is really focused around computational imaging and artificial intelligence, primarily in the context of radiology and pathology. So I want to talk about some of the implications of this in the context of precision medicine and mostly in the context of cancer or oncology. So this is my sort of brief conflict of interest disclosure slide. Very privileged to be working with many industry players where we are looking to translate some of the technologies that Dr. Shukla mentioned in terms of patents, working with some of these industry partners to really translate the technologies out into the clinic. For those of you who are on social media, I also have a Twitter profile. So if you like any of what you hear during the course of my talk and would like to follow along, I like to tweet about the work that's going on in our group. I tweet about other things as well, but if you'd like to follow along, that's my Twitter handle. So I want to set the stage a little bit for the clinical problem, the clinical context. And as you can see here on the slide, the lifetime probability of a man in the United States being diagnosed with some form of cancer is 1 in 2, or 50%. For women, that probability is only marginally higher. About 1 in 3 women will be diagnosed with some form of cancer in her lifetime, meaning that about 40% of the US population will be diagnosed with some form of cancer in his or her lifetime. Now, one of the challenges with that high diagnostic rate is, of course, well, what is the corresponding mortality rate? Well, it turns out that even though still a lot of people do die because of cancer every year, and chances are that probably in the next year, we're going to see even a greater increase in the number of cancer mortalities because of the pandemic, because people are not getting screened, and chances are that a lot of cancers are going to be found at a later stage. When I mentioned that about 40% of the population will be diagnosed with some form of cancer, when you look at the fact that the mortality is still, is high, it's about 600,000 deaths per year, there's still a big mismatch between the diagnostic incidence and the actual number of people dying because of cancer. And like I said, that's on account of a few different reasons. One is because we're doing a better job with screening for cancers, we're identifying the cancers early, we're using imaging in a far more aggressive way to identify the cancers early, and we also have better treatments to avail of, such as immunotherapy that got the Nobel Prize in Medicine in 2018. But there is also the very real fact that for a lot of cancers, we are over-treating and over-diagnosing a lot of cancers. And prostate cancer is a classic case in point, where 1 in 6 men in the US will be diagnosed with prostate cancer, but only 1 in 40 will die on account of it. And the problem with prostate cancer is that because most prostate cancers are fairly indolent, that is, they're not aggressive, they probably won't kill you, a lot of men could benefit from what is called active surveillance or watchful waiting, which means that they don't actually need very aggressive therapy. They don't need surgery. But the problem is that we still continue to treat a lot of these cancers very aggressively. A lot of men who don't need any sort of treatment because the cancer is growing so slowly, and they'll probably die with the cancer rather than on account of the cancer. A lot of these men don't need to be treated, but they still get surgery, they still get over-treated for very indolent cancers. And the problem is that over-treatment comes with a cost. It comes with a real cost to the patient because it can cause harm to the patient, it can cause complications, but it can also cause what is called financial toxicity. And in the US in particular, this is a big problem because in the US, you the way the health insurance is set up, it's different from the UK where you have the NHS. In the US, the problem is that a lot of patients had to end up paying out-of-pocket fees, out-of-pocket expenses. And so a large number of cancer patients in the US end up losing their life savings after a cancer diagnosis. Many of these patients end up losing their life savings within 1 year of that initial cancer diagnosis. And so this is a major, major issue. Overtreatment not only causes harm in terms of toxicity and complications to the patient, it also causes issues with regard to financial toxicity for the individual patients. So what I want to talk about today is with regard to developing better artificial intelligence tools for not just addressing the diagnostic problem, which is identifying the presence or absence of disease on imaging data. I also want to talk about the role that these tools can play for disease prognosis and predicting therapeutic response. So in other words, being able to predict disease outcome, predict disease progression, as well as identifying in advance of the therapy as to whether a patient is going to respond to the treatment or not going to respond to the treatment. And so it's really the development of these AI tools for prognosis and prediction of treatment response that ultimately will allow us to realize the promise of precision medicine. That is, being able to tailor a given therapy for a given patient based on their particular risk profile. And that's where AI and radiomics and computational imaging can play a significant role. Now, this is not to suggest that there aren't tools that already exist to address this problem. Probably the most famous tool that we're aware of is what is called Oncotype DX, which is a companion diagnostic tool that uses gene expression to really distinguish between patients who have a high risk of cancer and therefore have more aggressive cancer versus patients who have a less aggressive cancer. So this Oncotype DX assay, this molecular assay, really allows for distinguishing more aggressive patients who need chemotherapy from less aggressive patients who don't need chemotherapy. And this is a big deal because if you can distinguish patients who do need and do not need chemotherapy, then you can spare a lot of patients from toxicity. Because if you don't need to get chemotherapy and you can be cured with surgery and hormonal therapy alone, then that's a big, not only a cost saving, but obviously it avoids the deleterious side effects of toxicity of chemotherapy to the patient, right? So that's a big, big advantage, and these tests like Oncotype DX help address that. Now, one of the problems with Oncotype, like I said, is it is a molecular assay. It is a gene expression-based assay. And the problem with that is that it involves destroying the tissue, and it's also a very expensive test. These tests can cost something like $4,000 per patient. And also because of the intense heterogeneity of the disease, very often you end up sampling a part of the tumor to do the sampling of the genomics or the gene expression, and you may not really pick up the most aggressive component of the tumor when you're teasing off the tissue from the tumor sample. And so this is a challenge. So even though you spend all this money, you destroy the tissue and you get the Oncotype DX assay prediction, it may be that the actual risk assessment is not truly a reflection of the aggressiveness potential of that particular cancer. On the other hand, if you look at just routine tissue images, so any cancer patient who is suspected of cancer or has cancer will get a biopsy. And so with that tissue slide can be digitized, and you can create high-resolution images of the tissue samples, which can then be analyzed by the computer. And by analyzing the patterns with the computer, you can now, in a non-destructive way, start to describe the morphology of the disease in a highly quantitative and reproducible way. So, using image processing and computer vision, one can identify individual cancer cells on these pathology images. One can use network theory to start to look at the graphical arrangement and the network arrangement of the individual cells and convert this cellular network into a series of quantitative features that describes the spatial arrangement and the density and the architecture of the individual cells. One can also start to look at the textural patterns and the heterogeneity patterns of the individual tissue compartments, looking both at the stroma, looking at the epithelium. So different tissue compartments one can start to analyze in a very detailed and quantitative way. And so the advantage is that one can now convert this entire tissue slide image into quantitative features, into a quantitative signature that really reflects what the aggressiveness potential of the disease is. And this is the term that is sort of broadly described as pathomics. Now, about 15 years ago, when I got involved or got interested in the space of digital pathology or computational pathology, I had the good fortune of working with an oncologist, Sridhar Ganesan. And Sridhar saw the tools that we were developing for analysis of pathology images. And he said, Anant, look, you can develop these AI algorithms for analysis of pathology, but really from my perspective as a clinician, what I'm interested in is knowing who to treat with chemotherapy and who to avoid chemotherapy. And so working with Sridhar, we came up with this technology called Ibris, or the image-based risk score. And the main idea with Ibris was that you could take the tissue images, you could digitize those images, and then analyze them by using the image-based risk score. And so the advantage then was that you could now stratify these patients into those patients who had higher risk disease, who were going to benefit from chemotherapy, versus the patients who had lower risk disease and therefore could avoid chemotherapy altogether. Now, one of the questions is, well, how do you go about, what are the tools that you need to create this image-based risk score? Well, one of the critical things is the feature mining. So you've got to be able to extract the features. To extract the features, one has to be able to identify the individual cells, the individual nuclei in the images. And so one of the things that our group has been doing, like a number of other groups, has been using deep learning algorithms for identifying and segmenting individual nuclei. This is work we published a few years ago, where we used a stacked sparse autoencoder for nuclei detection, where we provided a series of different annotated examples or images to the network. The network was then able to learn the most appropriate representations and was then able to automatically identify the individual nuclei and the individual cells in these pathology images. And one of the things that we've also done is to really improve the efficiency of these algorithms. So you can see here on a very large image, this is about 40,000 pixels by 40,000 pixels. You can very rapidly go in and identify the individual cells, individual nuclei, and really create very detailed maps that show the location and the position of the individual types of cells, the cancer nuclei, the lymphocytes, on these very, very large pathology images. Now, the advantage is that not only can you do the segmentation and detection of these individual cells, you can also start to classify the different types of cells. So this is where we've used networks like generative adversarial networks, or GANs, to distinguish between cancer nuclei from lymphocytes, so cancer cells from the immune cells, and you can see them colored here by these GANs with different colors. So the beauty is that you can identify these different cell types. You can also identify the different tissue compartments. So like I said, there is the stroma, there is the epithelium. These are the different tissue compartments that comprise the tumor. And so what we've been very interested in has been to really find different cell types and different tissue compartments and look at the spatial arrangement and the spatial architecture of the different cell types within the individual tissue compartments. And by analyzing the architecture and the arrangement of these different cell types, we can now start to convert these images into quantitative features and quantitative measurements that can describe the morphology of the disease in a highly quantitative way. Now, a few years ago, we were using deep learning to go in and segment the individual cells. But one of the questions we also thought of asking was, could you just feed a number of images to a deep neural network and see whether you could directly predict what the outcome for a patient might be? And so we started this study where we're using patients who had heart failure. And so we were looking at just the H&E tissue biopsy images in these patients. And we said, well, could you train an algorithm by knowing whether these patients had heart failure or not? Could we now just develop an algorithm that could predict the likelihood of heart failure from the patterns that the network was able to learn from the images directly? So we did a study where we had about 100 patients that constituted the training set. We had another 100 patients for the validation set. And we also had two pathologists who went in and looked at the predictions of the individual biopsies. So they were also used for sort of a comparative study of the neural network. And what happened was when we applied the neural network trained on 100 images to the validation set, the neural network came back with an area under the curve, ROC curve, of 0.97. So 97% accuracy or performance in correctly distinguishing heart failure from non-heart failure based on these heart biopsy images. On the other hand, the two pathologists only had an average area under the curve of 74%. So big difference between what the pathologists were able to do and what the neural network was able to do. And this was, really thrilling. We were able to get a lot of publicity out of this, get a lot of visibility out of this paper that was published. However, when a few months later we repeated the study with additional patients, we found that the neural network dropped significantly. It went from 97% to about 75%. And the only thing that had changed was that the patients had been scanned on the same scanner. The slides had been scanned on the same scanner to generate the images. The only difference was that the scanner had had a software upgrade. And because of that, the distribution of the intensities, the color of the images had changed in a very subtle way. And because it had changed in a very subtle way, the neural network now was not able to perform at the same level that it was performing prior to the initial sort of software upgrade being applied to the scanner. So compared to the first batch of images where it was about 97%, the second batch fell dramatically to about 75%. So that really suggests that there's a great deal of sensitivity associated with these neural networks. And one really has to approach these methodologies with a great deal of caution. And way smarter people than me have talked about this very eloquently. Dr. Michael Jordan at the University of California, Berkeley, who's extremely well known for his work in machine learning, has talked about this, right? These brute force algorithms like deep learning, one puts too much faith into these algorithms, right? And that we have to be more skeptical about what these algorithms are truly learning. And here's another example that I like to use very often. This is an example that's actually from Samir Singh at the University of California, Irvine. And what Samir and his student had done was to train a neural network to distinguish huskies from wolves from images that they found on the internet. And they trained a deep learning algorithm and applied it to distinguishing huskies from wolves and found that the network did a very good job, that they were able to get a performance of something like 99%. But Samir, who realized that the problem is very difficult and that something had to be wrong because huskies and wolves look very similar to each other, right? And so when they went in and analyzed these images more, they found that the huskies were always against a white background. There was always snow in the background, and that was really what was distinguishing the husky from the wolf. The features weren't coming from the face of the animal. The features were coming from the background, right? And so that was fundamentally what was driving the prediction. And so our group, therefore, is really focused on handcrafted features, and which we believe provides an intuitive alternative to deep learning, right? So what we have really focused on are how can we use image processing, computer vision to analyze these images to extract features that reflect something about the disease, that tell us something about the biology of the disease. Now, one of the things that we've been very interested in is looking at what is called collagen. So collagen is this material that's present in tissue that really provides the binding mechanism for the tissue. It holds it together. But it turns out that when you've got cancer in the tissue, that the cancer grows aggressively, and then the collagen fibers start to change in their orientation. They get distorted as the cancer grows. And so our hypothesis was that if you can identify and segment out the collagen and look at the orientation of the collagen fibers, maybe there is differences that we can identify between patients who have short-term survival, that is, they have more aggressive cancers, that's what you see here on the left, And on the right, you've got patients with long-term survival. So these are patients who are likely to have better outcome and may not need chemotherapy. So we did a study which we have just about to submit for consideration for publication, where we looked at the orientation of the collagen fibers and found that in patients who tended to have short-term survival, the orientation of collagen fibers was more structured. So the fiber orientations were all in the same direction. In patients who had long-term survival, that is, they did better, the collagen fibers were more disordered. They were more chaotically arranged. And because they were more chaotically arranged, there were significant differences between the short-term survivors and the long-term survivors in terms of their final outcome prediction. So what you see here are what are called Kaplan-Meier curves. These are what are called survival curves or survival statistics that one can glean. And so if you see these red curves and blue curves, what it means is that the patients who have poor survival can really be distinguished from patients who have better survival. That's in the blue curve. And if there is a difference here, and it's a statistically significant difference, it means that your classifier or your features are able to tell apart between the patients who have poor survival from good survival. And so you can see here that the feature of collagen fiber orientation did a really good job in distinguishing between patients who had poor survival from good survival. And you can see here with both of these cohorts. And so here's an example where we used not some black box feature, but used a feature related to collagen fiber, which we know is critical for pathology, for the outcome of these patients. So we were able to model a meaningful feature, meaningful attribute and connect it to outcome. Now, there are other features as well. I mean, there are simpler features like the shape of the individual nuclei that we've also looked at. We've looked at the orientation of the individual nuclei. So this is another example here of looking at not just nuclear shape, but the orientation of individual nuclei as well, and shown that the combination of nuclear shape features and nuclear orientation features can really distinguish between patients with long-term survival and short-term survival. And so why is this important, right? So remember I talked about the Oncotype DX assay, which is a gene expression-based assay. And this gene expression assay, like I said, is very expensive. It's $4,600. But the Ibris assay, the image-based risk score, remember it only uses an image. And because it only uses an image, it's much lower in cost compared to the gene expression-based assay. It's very, very quick compared to the Oncotype DX assay because you can return the results in a matter of minutes compared to shipping the tissue back and forth. You're not destroying any tissue with the Oncotype. With the Ibris, it's a digitized image. And of course, you can have a worldwide impact because the images can be electronically sent over for analysis from anywhere in the world. And so, in other words, you have an inexpensive, fast, reliable, accessible prognostic breast cancer test that's priceless. For everything else, there's MasterCard. So that's my little joke there. One of the other things that we've also been doing has been how can we combine the image score or the image predictor with the molecular predictor. And so we've shown that combining the image features with a molecular score allows for even better prognosis and even better outcome prediction. So we did the study where we took the Oncotype DX, the gene expression assay, combined it with the image features, and showed that the combination resulted in improving the performance of the image of the Oncotype assay, the $4,000 test, by about 20%. So we were able to identify 20% more truly low-risk breast cancer patients who could avoid chemotherapy, right? So that's a big deal because this expensive test, even though it's so expensive, still misses a lot of breast cancer patients who could avoid chemotherapy. And so we showed in the study that bringing in the image-based features, that you could improve the performance of this assay, this gene expression assay, by about 20%. But it's not just in breast cancer. We've shown this work applies to other types of cancers. This is in the context of early-stage lung adenocarcinomas, where we've demonstrated that we can identify patients who are likely to have recurrence, and shown that these features that relate to the heterogeneity of the shape and the texture and the spatial arrangement of these cells it's quite different between patients who are low risk and high risk. And again, you can see that the curves that I talked about, the survival curves, and you can see here, and we can really separate out quite nicely between the patients who have good survival versus the patients who have poor survival. One of the other areas we've been looking at is prostate cancer, where we focused on looking at the shapes and the arrangement of the individual glands that are in the prostate pathology images. One of the questions we've been asking is, can we look at the shape of the individual glands from the prostate cancer images to predict which patients will have recurrence of cancer, in which case the cancer will come back after surgery. And so we've demonstrated in a pretty large study, almost 1,000 patients, that these features are prognostic, and we can identify which patients are likely to have a recurrence of the disease versus not have a recurrence of the disease. But what is also really interesting is we were able to compare our image features with a gene expression-based assay. So just like I gave the example with the Oncotype DX in breast cancer, there is a gene expression assay called Decipher for prostate cancer. That's also expensive. That's used for identifying which patients will have cancer recurrence, and showed that the image features were just as good as the gene expression assay. But what was interesting also is that when we combine the image features with a couple of clinical parameters, just 2 clinical parameters, we did almost twice as well as the gene expression assay. So just by using the images alone, the image features, combining it with 2 clinical parameters, we could get almost twice the performance of the very expensive and tissue-destructive molecular assay. This also has implications for other types of cancers. Oral cavity cancer is obviously a big problem in India. It's I think you probably see more oral cavity cancers in India compared to, I think, pretty much any other part of the world. Clearly, that has a lot of to do with the tobacco chewing and the betel leaves, the tobacco and the pan that I'm sure a lot of people on the call today are familiar with, and that is a major reason for oral cavity cancers. One of the things that we've been asking is, can we identify using our computer vision image analysis approaches which oral cancers are more aggressive and will progress and need the very aggressive treatment versus the oral cancers that are less aggressive, that could benefit from less aggressive treatment where you can avoid chemotherapy or radiation therapy. So this is data that we published a couple of years ago. More recently, we've had some very exciting data where we've actually demonstrated that in a small cohort of patients, that we can identify the patients who could benefit from surgery alone. So this is the blue line where patients do well, so they can just get surgery. But then we also identified patients who do poorly, and these are the patients who really need the chemotherapy or radiation therapy. That means they need surgery plus something else. So surgery is sort of the base option where it's the minimal thing that you have to do. And so the patients on this blue line are the ones that can get surgery alone. But the ones in red are the ones who probably will need additional therapy over and above surgery. That is, they need radiation therapy or chemotherapy. So again, just using these image features alone, we were able to distinguish between more aggressive and less aggressive cancers. Another area we've been looking at are what are called oropharyngeal cancers. These are head and neck cancers. And this was sort of an interesting study where we combined deep learning with handcrafted features. So one of the things that we've recognized for a long time is that multinucleation, which is a particular type of finding in pathology images where these multiple different nuclei stick together, this multinucleation is something that has to be, is known to be a hallmark of poor prognosis. So if you see a lot of these cells sticking together, we know that they represent patients who are going to do poorly. And so we did a large study of almost 1,000 patients where we used deep learning to first go and identify the individual multinucleation events, and then demonstrated that using this multinucleation alone, that we were able to distinguish between patients who had good survival versus poor survival. So we were able to really distinguish between those patients. Now, one of the other things that we've also been doing has been looking at the spatial arrangement of the immune cells. Now, we know that immune cells are very strongly implicated in a number of different cancers. We know that these immune cells are really critical for identifying which patients are going to do poorly versus which patients are going to do well. Now, most people who are studying this problem of immune cells are looking at just trying to identify the number of immune cells. We took a different approach. We said, could we use deep learning first to identify immune cells and then cancer cells on tissue images? And we used a clustering algorithm where, and deep learning, where we identified clusters of immune cells. So these are in green. And we also identified the clusters of cancer cells, which are in red. And now using spatial statistics, using network theory, using graph algorithms, we were able to convert these co-arrangement patterns of immune cells and cancer cells into quantitative features that told us about the relative co-arrangement of the immune cells with respect to the cancer cells, and found that these features could distinguish between patients who had recurrence versus patients who did not have recurrence. So there was a strong separation between patients who did poorly versus patients who did well. Now, one of the other things that we've also we've been very interested in, and that I mentioned at the start of the talk, is that we're really looking at applying these approaches to identify response to therapy. So I talked about immunotherapy, which is a new therapy that a lot of you may be familiar with, that has really changed the landscape of how we approach cancer today. It got the Nobel Prize in 2018 in medicine, and is giving much better responses to chemotherapy in a lot of cancer patients. The problem is it only works 20% of the time. So if you apply this treatment to 5 patients, only 1 will respond. The problem is that it's very expensive. It's something like $200,000 per patient per year. So that is, if you treat 5 patients, it's $1 million, out of which $800,000 is going down the drain because of ineffective treatment. So one of the things that we've been doing has been to look at this co-arrangement of immune cells and cancer cells to try to distinguish responders from non-responders to immunotherapy. And one of the things that we've demonstrated very nicely is that if you look at lung cancer patients, that these features of the co-arrangement of immune cells and cancer cells really can distinguish between patients who have poor survival versus patients who have good survival. And so we've shown this quite nicely across multiple different cohorts. We've shown this quite nicely across different sites, and in fact, even different therapeutic agents. So for different types of immunotherapy drugs, we've demonstrated that these patterns are able to distinguish patients who do well versus the patients who do poorly. We've shown this in other types of cancers as well. Ovarian cancer is another area where this arrangement between immune cells and cancer cells has been shown to be prognostic. Again, you can see with the separation of the red and the blue curves that we can tell apart patients who do well versus patients who do poorly. Also endometrial and cervical cancers, other types of cancers, again, with pretty large studies, we've demonstrated that you can really tell apart which patients are going to do poorly versus which patients are going to do well. And then I talked about immunotherapy, and I showed the work for lung cancers. It turns out that even in the context of patients with gynecologic cancers, so patients are treated for ovarian cancer or endometrial cancer or cervical cancer, that we can tell apart which patients are going to respond or benefit from immunotherapy versus not going to respond to immunotherapy. Now, obviously, I can't give a talk around AI and not talk about COVID. Of course, it is the #1 thing that occupies the entire world today. It completely upended our life, in 2020. And so one of the things we've been doing has been to look to understand with computer vision and image processing, what are the differences in the immune patterns of COVID-19 versus H1N1? And so this is a preliminary study that we've done. This has just been submitted, or actually in the process of being submitted for publication, where we found that the immune patterns in COVID-19 patients and H1N1 patients is very different. And we find that in H1N1 patients, the immune cells form larger clusters, denser clusters. In COVID-19 patients, they're much smaller, fragmented clusters. And so again, this is all just analysis of the tissue images of patients who died either because of COVID-19 or because of H1N1. One of the other questions that we've been asking is, well, should we only be looking at the cancer? Should we only look at features that come from the tumor? And in fact, in work that we published a few years ago, we showed that features that come from outside the tumor in the apparently normal areas also tend to have features or patterns that can tell us a lot about how these patients are going to do, whether they're going to have a recurrence or whether they're not going to have a recurrence. One of the other questions we're also asking, this is work we just published recently, is that the features of the cancer can actually look very different between different populations. So if you look at patients who are African American or blacks versus Caucasian American or whites, the features of the cancer can be quite different. And so that means that the kind of machine learning models you create for, for patients of one particular population or ethnicity might need to be different from the model that you build for another ethnicity. So, and we also have a collaboration ongoing right now with the Tata Cancer Center in Mumbai, where we're looking at Indian women with breast cancer and finding that the features of Indian women with breast cancer seems to be quite different from the features for white women or Caucasian American women in the United States. So different populations might have different features associated with the disease that needs to be taken into account when you're creating these prognostic predictive models. Let's skip over this slide in the interest of time. I talked about the work we're doing on pathology, but one of the other things that we're also working on is radiology, or what is called radiomics. And one of the questions we've been trying to address in this particular context is trying to distinguish between brain tumor patients who get treated with chemotherapy and radiation. So specifically, we've been trying to identify which patients with brain tumors will have a recurrence of the cancer after treatment. And at the same time, try to distinguish which of these patients will just have a treatment effect. So a lot of these patients that get chemotherapy and radiation will have what is called radiation necrosis. Radiation necrosis is completely benign, it's not cancerous, but the problem is that radiation necrosis looks very similar to cancer recurrence. So when you look at the MRI scans for these patients, a radiologist really can't tell apart what is radiation necrosis from what is cancer recurrence. And so a lot of these patients have to end up getting a biopsy and to try to figure out whether it's the cancer come back or whether it's an effect of the treatment. What we've developed is a new radiomics approach that uses image processing computer vision tools to first find at a pixel by pixel level the dominant edge in the image. And then by using the concept of entropy, we're able to capture the disorder in the orientations of the pixel level edges. So what that allows us to do is to convert the MRI scan into entropy maps, right? And so if you think about these colors, the red colors represent high entropy. And so the high entropy is areas where the orientations of the edges are very discordant with respect to each other. They're very disordered. The blue represents areas where the orientations are more or less in the same direction, where they are more structured, more homogeneously oriented. And so now you can really see that there's a big difference between the patients who have recurrence of the cancer, you can see that here, versus radiation necrosis, where you see much more homogeneity in the morphology compared to the patients who have the recurrent tumor. So by using these entropy maps, now a radiologist can distinguish patients who have a recurrence of the tumor versus patients who have just radiation necrosis. And this is work that was led by Dr. Pallavi Tiwari, who's a junior faculty in biomedical engineering, and a former graduate student in our group, Pradeep Prasanna, who's now an assistant professor at Stony Brook University in New York. So in another related study we just published recently, we found that these radiomic features or entropy features in brain tumors could also tell us about survival, could also tell us as to which patients who had long-term survival versus short-term survival. And interestingly, that the features that were prognostic of survival didn't just come from the cancer region, but also came from outside the cancer region. And so one of the other things we also did in the study was to connect the radiomic features of the tumor and outside the tumor with the molecular features of the tumor. So we were able to look at different pathways that the tumor was implicated in and showed the strong association between the radiomic features as well as these molecular pathways of the tumor. We've taken a similar approach and applied it to prostate cancer and showed that in prostate cancer as well, that these radiomic features are able to distinguish between patients who have recurrence of the cancer versus patients who don't have recurrence, and have also shown that these radiomic features are strongly implicated with the molecular association of the tumor. So the Decipher risk score that I talked about previously, we showed is actually strongly connected with these radiomic or entropy features we're getting from MRI scans of the prostate. One of the other features we've been looking at is the shape of the tumor and the shape of the prostate, and found that in patients who have recurrence of prostate cancer after surgery versus patients who don't have recurrence, they look quite different. And so we created an atlas-based approach where we looked at patients who did have recurrence and created one atlas. We had another cohort of patients where we constructed another atlas and looked at the difference in the shape and the distension of the prostate capsule between patients who did and did not have recurrence of the cancer, and found that the shape and the distension was quite different between patients who did and did not have biochemical recurrence after surgery. So this is, this again goes back to the point that I made, that you want to really look for, non-black box features, right? So this is intriguing because now you have an appreciation for what the feature means. You've got more interpretability associated with your feature. And you're also able to identify where the features are truly different between patients who have recurrence versus don't have recurrence. We've taken this approach to other cancers. This is work that we're doing in rectal cancers, where we've shown that the entropy features are also able to predict response, treatment response in rectal cancers from MRI scans. We've shown that you can also predict response in breast cancers to chemotherapy, so figuring out which patients will respond to chemotherapy versus not respond to chemotherapy. Again, this is off an MRI scan. So all this work that I'm talking about now is based on MRI and CT scans. The first part of my talk was all based on tissue images. Now we're talking about radiographic scans. And so here, in work we published a couple of years ago, we showed that the features, again, outside the tumor, the entropy features outside the tumor, could really tell us as to which patients were going to respond to chemotherapy versus patients who were not going to respond to chemotherapy. One of the other things that we've also done is shown the association between the image features and what's happening at a cellular level. So we were able to get biopsies of these breast cancer patients outside the tumor and showed that there was a strong association between the entropy features from the MRI, which you're seeing here in the left, with the tissue images, which you're seeing here in the middle. And so it turns out that the spatial arrangement of the immune cells that we were able to identify with deep learning on the biopsies was strongly correlated with the entropy features outside the tumor on the MRI scan. So strong association between what you're seeing on radiology with what we were seeing on the pathology. And this was important because this could explain, again, why we're seeing the particular features we are on the MRI scans. Another area we've been working on is in lung cancer, and shown, this is work done by Pranjal Vaidya, who's a graduate student in our group, and Pranjal has shown very nicely that the radiomic features from CT scans, again, inside and outside the tumor, could really tell us not just about the survival characteristics of these patients, but can also tell us as to which patients are going to respond to chemotherapy or going to need chemotherapy versus patients who are not going to need chemotherapy. I talked about immunotherapy previously and showed that from the tissue images, we can predict who is going to respond to immunotherapy versus not respond to immunotherapy. We've now demonstrated that from CT scans, again, looking at features from CT scans, we can also distinguish between patients who are going to respond to immunotherapy versus patients who are not going to respond to immunotherapy. Skip over the slide in the interest of time. One of the other things we've also been looking at, like I mentioned, a lot of interest in our group in looking at non-intuitive, more intuitive features. So rather than looking at the tumor, our group has also been looking at the vessels that feed the tumor. So specifically looking at the twistedness and the shape of the vessels that feed the tumor, and found that in patients who respond to chemotherapy, the vessels tend to be much smoother. In patients who don't respond to chemotherapy, the vessels tend to be more twisted, more convoluted, and significant differences in the morphology and the shape of the vessels between tumors that don't respond versus the patients that do respond to chemotherapy. Similarly, Mehdi Alilu, who's a research faculty member in our group, has shown that these features are also different between patients who respond to immunotherapy versus patients who don't respond to immunotherapy. So patients who respond, the vessels are less twisted, less convoluted. Patients who don't respond tend to have a much more twisted, much more convoluted vasculature associated with them. Finally, we've also shown that the vessel tortuosity tends to change over treatment. So if you look at the baseline, responders tend to have much smoother vasculature compared to non-responders. But then as the treatment progresses, you find that patients who respond, the vasculature of the tumor tends to become even less twisted. But in patients who don't respond, the vessels tend to become even more twisted and more convoluted. Finally, I just want to talk about some of the work that we're doing in a collaboration with Dr. Jonathan Liu, who's a biomedical engineer at the University of Washington, Seattle. And Dr. Jonathan Liu's group has pioneered a new microscopy imaging technique called light sheet microscopy that allows for three-dimensional imaging of tissue scans, of tissue biopsies and tissue pathology. And so what we've started to do now has been to apply 3D segmentation approaches to capture the individual glands and the individual cells on these pathology images. And looking here, you can tell just immediately at the 3D shape of the gland that in cancers, the shape is more irregular compared to the benign or non-cancer regions where the shape is much smoother, much more organized. So this is something that's really exciting that we're pursuing, looking at 3D features of 3D pathology compared to 2D features for not just diagnosis, but also prognosis and predicting therapeutic response. So with that, let me conclude my talk. So hopefully what I've conveyed is that computational analytics with routine imaging can help address questions in precision medicine, specifically prognosis and predicting response to therapy. One very important component here is the low-cost aspect. And that's really critical when you want to have global impact, right? Particularly in low and middle income countries like India, South Asia, China, Africa, Latin America, but cost is very critical. So being able to use AI with routinely acquired data becomes very, very critical. Hopefully what I've conveyed is the need to not just look at black box or opaque approaches like with deep learning. Deep learning, I think, has its advantages, but deep learning is not something you can throw at every problem. And really, there's a need for more interpretable approaches, more interpretable solutions, and also identifying and establishing the underlying basis of your approaches. So not just saying that you've got a predictor or classifier, but taking it one step further, trying to understand from a cellular level, from a molecular level, why is your predictor working? What is driving the predictor? So really establishing that molecular basis is going to be important for clinical adoption. I just have to acknowledge the team, the amazing students and postdocs and faculty members that I have the pleasure and honor of working with on a daily basis. And so this is work that's really done by them. I'm just happen to be presenting on their behalf. And of course, we always have to thank the funding agencies who make the work possible and provide the funding for the work. And so again, with that, I'd like to thank all of you for your kind attention. Again, for questions that I may not be able to address now, right after the talk, please feel free to connect with me. Again, like I said, this is my Twitter handle. For those of you on Twitter, please feel free to reach out or do a follow, and feel free to shoot me a message. Thank you very much.
Yeah.
Happy to take questions.
Yeah, Professor George, are you? Hello?
Hello?
Yeah. You are audible. Professor George, yeah.
Dr. Ananda, hi, I'm George Yakos. I have two questions.
Yeah.
First of all, your talk was very interesting, very informative, and we appreciate your effort on this research direction. I have a few questions. What is the impact of F18, let's say, PET tomography on the prognostic grade assessment, specifically sarcomas? You know, they apply this technology at Mayo Clinic, but have you any idea this could provide metabolic information of cancer?
Yeah. No, I think that's a fantastic question. Thank you, George, for asking that question. One of the things that we have to be aware of is trying to understand where and when these different imaging modalities can play a role. Now, PET, I think, is very powerful. You talk about FLT PET, which we've done some work on before. Obviously, glucose imaging with FDG PET is very, very critical. And PET really has started to play a significant role in prediction of treatment response, in predicting outcome, in identifying metastasis, for instance. So one thing that has to be considered with these different modalities is where can they really play a role? And it depends on the kind of question you're asking. If you're looking at trying to do disease diagnosis, it may not be the kind of modality that you want to use because PET is also expensive. But if you're looking at trying to identify the likelihood of metastasis, if you're trying to figure out which patients are going to have, spread of the disease, then I think in that context, PET imaging becomes useful. But if you're doing diagnosis, then maybe you want to look at MRI, you want to look at CT where appropriate. So in sarcomas, for instance, we started a project, you mentioned sarcomas, where in fact we're looking at MRIs. And one of the problems with MRI is the diagnostic problem in really distinguishing between sarcomas and benign presentations. And so that's where we're using AI with MRI scans. So thank you for bringing that up. It's a very critical question. It really, in my opinion, in my humble opinion, it's a question of the particular clinical question that you're asking and making sure that the modality that you're looking at can help address that question. But part of the equation, like I said, is also balancing out the cost aspect, right? Because all of these things are expensive. PET imaging can run from $2,000 to $3,000 for a scan, right? CT can cost about $100. So one of the questions that you have to ask with getting a PET scan is, what is the added value? I think certainly in the context of prognosis and predicting metastasis, it clearly has significant role. Thank you for asking that question.
Thank you. I have other two questions, if you don't mind.
Sure. Yeah.
One question is, you talk about at the end about the light sheet microscopy. To my understanding, this should be equivalent to confocal microscopy, where you take different planes, and after you find volume information, you have scatter ejection from different planes. Or how stands with OCT microscopy? Somebody can apply OCT, which would save the job.
Yeah, so I think that one thing about light sheet microscopy is the exquisite detail that you can get, the exquisite granularity that you can get that OCT is simply not able to provide. OCT is fantastic, but with the light sheet microscopy, I've just been blown away by the quality of these images that you're getting. I mean, the images that I showed at the end there represent simulated images. So these are digital images where the H&E colors have been simulated, have been overlaid on top of those simulated images. It looks almost indistinguishable from something that has been created in a pathology lab. You can't tell the difference. It's unbelievable. I mean, just the fidelity of the light sheet microscopy images is just unprecedented. So I think that the granularity and the resolution of morphology that you can see is really significantly better than anything that I've seen with OCT. It does sort of use the concept idea in a sense. It's like the computed, it's like a tomographic scan, right? So in a way where you've got multiple of these light projections, and then you're doing a reconstruction, a reconstruction, a 3D image. I'm not the developer of the technology myself, so I would have to direct you to Jonathan Liu, who developed the light sheet microscopy imaging platform. But just in terms of the data and the quality of these images, having played with a lot of these images, it is just phenomenal. The kind of three-dimensional features that we're able to prize out from it is just volumes above anything that you can get from 2D pathology images. So really, really impressive.
Thank you. Thank you.
Thank you. Thank you. Now, any question from anyone?
I have another question. I was asked due to the interest. Okay, because let's start talking about the initial part of your talk on the morphology of cancer tissue. With the collagen formation or collagen, degrading, etc.
Yeah.
Morphological information. Now, since I work on this area for many years, I'm choosing a different approach, the interrogation of light, the wavelength. So we're using near infrared instead of visible. I guess your images that you're getting, you're using common visible light, correct? So my question is, did you explore the feasibility of using near infrared or a specific wavelength rather using general visible light to interrogate and catch more features?
Fantastic question. Thank you for asking that question, George. So one of the things that we've been starting to have conversations about is using these specific wavelengths that can really accentuate collagen. I've been working with a couple of people who really are pioneers in this space. One is Dr. Kevin Elisieri at the University of Wisconsin-Madison, who's developed really innovative ways of imaging collagen using these dedicated specific wavelengths. And the other person that I know who's working in this space, there are a number of people, but one person that I'm working with is Dr. Richard Levinson, who's at the University of California, Davis, and he's been using this approach. Now, one of the things that we tried to do was to use the routinely acquired data. So we wanted to look at just the standard H&E images and see what we could pull from that. There's no doubt that if you image with these more dedicated collagen imaging modalities, that you could probably get even better fidelity, you could get even better characterization of the collagen fibers. It could do even, possibly more robust features, more, better features from dedicated collagen fiber imaging approaches. But we're finding that even with the H&E, which doesn't require any specialized imaging, we're still putting out features that are strongly prognostic. So to me that's very satisfying, because it means that even from the baseline data that we have, we're still able to pull out a prognostic signal. Now, once we start to layer on additional imaging features, imaging parameters, we might be able to do better. But it's very satisfying that even with the standard H&E, there is a prognostic signal that we're able to prize out.
Thank you very much. Thank you. I notice you're using in your presentation stained tissue. Why you didn't try with stainless tissue, where you can introduce less damage to the tissue, can be more inexpensive?
That is exactly the technology that Jonathan is using. So the light sheet microscopy, well, there are two parts to this. Let me try to address this question in a couple of different ways. So one is that for routine pathologic diagnosis, all the slides are ultimately going to be stained with hematoxylin eosin, H&E, hematoxylin eosin. It's a standard rubric for pathologic diagnosis. So any tissue or biopsy that is evaluated by a pathologist is going to have an H&E stain. So one of the things that we've been doing is, let us look to use what is already being acquired as part of clinical protocol, clinical practice. So that is one piece. The second piece is regarding stainless. I think that's a great idea. In fact, the light sheet microscopy work I showed at the end, Jonathan Liu's group actually uses this unstained tissue. So they use unstained tissue blocks to do the imaging, and the colors that you see are actually layered on by simulation. So it's actually using machine learning to simulate the colors of what the hematoxin eosin might look like. So, you know, I think that in certain scenarios, using the unstained actually has major advantages, like with light sheet microscopy. At the same time, a lot of retrospective data that exists, a lot of digitized images, slides that exist are stained with H&E. And so by leveraging those H&E slides and H&E slide images, it provides a huge, you know, resource of data for training and validation.
I see. If you can send me at the end your email, I can send you some papers on our work on stainless. Stainless pathology, yeah. Since 2009, you know, we're working on this direction. So I would be glad to send you and get some, you know.
Yeah, absolutely. I think Dr. Singh has my contact details. I'm sure.
Thank you.
I will share with Professor George. Definitely.
Thank you. Very interesting presentation. Thank you very much.
Yeah.
Absolutely.
Thanks to Professor. I mean, it's very nice. The discussion between Professor Anand and Professor George becomes another presentation. So it's very informative discussion, and we would like to thanks both of you and all my participants. So today we are now going to close the session.
Thank you. Thank you very much.
Thank you. Again, thank you to all of you. Thanks, Professor George and Nandeba.
Thank you. Bye.
Namaste.
Namaste.
Namaste. Hello, Professor George. Hello. Professor George.